This week in lab, we are going to cover a lot of different kinds of stem cuttings. And to do that, first we need to understand the different parts of a stem and how we utilize those. So this would be a stem. Stems have nodes. The nodes are either where there's an axillary bud or a terminal bud. The terminal bud is always the one at the tip. The axillary buds are the ones that are in the other portions of the stem. Each axillary bud is typically associated with a leaf, but not always. And then you also have an internode. The internode is just the space between two nodes. One entire repeating segment that contains both a node and an internode is called a phytomere. And knowing how many phytomeres we have is going to help us with figuring out how many we need to keep in one stem cutting to have some of these nodes be under the soil surface and some of them be above the soil surface for our rooting. In experiment number 1, we are going to see if there's a difference in the number of leaves that you leave on your cutting and how successful your rooting is. So we're going to do this with chrysanthemums, and our 3 treatments are going to be, we're going to leave 3 leaves, we're going to leave 1 leaf, or we're not going to leave any leaves at all. And then we're going to see what the difference is in rooting success based on the number of leaves that we've left on our cuttings. In experiment number 2, we're going to look at the effect of cut location, where we cut on our stem and its rooting success. So we're going to cut right above a node, which is going to leave us a long internode piece that goes below the soil. And we're going to cut right below a node so that this node actually goes below the soil. And we'll see which one of those has better rooting success. And then also we'll make some observations about where the roots come out of a stem, just in general in an herbaceous plant. The plant that we're going to do this with is apple mint, Mentha suaveolens. The next experiment is going to be to determine if there's a difference in soil media and the water holding or airspace capacity that each of those has. And we're going to use 2 different kinds of media. The first is going to be called vermiculite, which is kind of like a puffed mica type rock. These are typically pretty big pieces, and there's a lot of air/water space in between of these. The thing that we're going to compare it to is regular potting mix, which contains some perlite and then some coconut coir and some shredded bark and things. And it's going to have it's going to hold a lot more water and have a lot less airspace in there. And we're just going to see if there's an advantage to one over the other. Sometimes your potting mix can be a little bit too heavy, and so your cuttings will rot before they root, and that might be an advantage to having bigger airspace, but we'll see which one works out better. And for this experiment, we are going to use Mexican sage. In experiment number 4, we're going to see if cutting type has an effect on rooting success, and we're going to do this with a variegated hydrangea, and we're going to do 2 different cutting types, terminal and subterminal. For a terminal cutting, you're going to leave that terminal bud or apical meristem intact, and so we're going to see if having this intact is changing the hormone structure so that it doesn't want to root and tries to just keep growing shoots instead. And so we'll make cuttings of both of those and see what happens. And then lastly, our final experiment of the week is going to be to determine the effect of rooting success based on rooting hormone type. We're going to use a control, a product called root juice, which is sold as an organic rooting compound, and Bontone 2, which the active ingredient in that is indole 3-butyric acid, or IBA. And then we'll have our control. We're going to do this with 2 different plant types. The first is a cherry plum, which is a really good commercial rootstock for grafting plums and peaches and nectarines and almonds. And then we're also going to do it with a rose of Sharon. Both of these are softwood cuttings that we're going to be doing, and we'll just see what happens. Okay, so that's our overview for today. Now we're going to go through and watch some videos of these experiments in progress, and then we have some observational exercises and some observations for you guys to make from previous experiments.